This is Microbiology Lecture 4. This is going to correspond to the Bauman text Chapter 4 and to the tutorial text Chapters 3 and Chapter 10. We're going to start looking at microscopy. With microscopy, you want to refresh yourself with the units of measure from the metric system. When we talk about 1 centimeter, it means 1/100th or 0.01 meter. 1 millimeter is 1/1000th or 0.001 meter. 1 micrometer is 1/1,000,000th, 0.0000001 meter, and then we have the nanometer that is 1/1000,000th. Microscopy uses light or electrons to magnify objects. We do this a lot with microbiology because we're looking at cells, and the vast majority of cells are not visible to the naked eye. Looking at the microscope, this is just a general picture of a microscope. There is quite a bit of variation in the different models of microscopes, but they're all going to have some of the same features. So knobs may be in slightly different areas. You may have on-off buttons in slightly different areas, but they're all going to have the series of lenses, the stage, and the principle of how they work is the same. In looking at general microscopy, there's a few things to consider. The wavelength is the distance between the two corresponding parts of the wave. Visible light is 400 nanometers to 650 nanometers from violet to red, with white falling at about 550 nanometers. Magnification is the apparent size of an object. It's indicated by a number, and then we use the letter X. It's the result of refraction created by a curved lens. The magnetic fields will refract electrons. Resolution is the ability to distinguish between two objects that are close together. Modern microscope resolution is about 0.2 microns, or micrometers. This actually depends on the wavelength of light, or the electrons, and the numerical aperture. The numerical aperture is the ability of a lens to gather light. We have a resolution formula that will tell you the resolution distance. It's equal to 0.61 times the wavelength, divided by the numerical aperture. The wavelength is often going to be your limiting factor in how good of resolution you can get. Contrast is the difference in intensity between two objects. This will improve with using stain. Contrast improves the resolution, so most of the time we will stain things. It's fairly easy to stain things, and you get a much better view underneath the microscope. Light microscopy is what we will be using. The bright field is the most common. The background or the field is illuminated. With a simple microscope, you have just a single magnifying lens. This would be what we would consider a magnifying glass. This is what Leeuwenhoek worked with in 1673. Compound microscopes are going to use a series of lenses for magnification. This idea was used as early as 1590 by Galileo, but it was 1830 before compound lenses could exceed the clarity of a simple lens. The refractive index is the measure of light bending ability of a medium. You can change this by staining, and this will give sharper contrast to the image. Your objective lens is going to be the lens immediately above the object being magnified. Your ocular lens is the one that's closest to the eye. It will typically magnify 10 times. Your objective lens will vary in its magnification depending on which one you slide in. The total magnification is determined by multiplying the magnification of the objective lens times that of the ocular lens. Immersion oil is used only on oil lenses. This preserves the deflection of the light rays at the highest magnification, so it will improve the resolving power of the lens. It is important that the immersion oil is only used on the lenses that are designed for oil, otherwise it can ruin the other lenses. Your illuminator is the light source, and the condenser is going to direct light through the specimen using mirrors and prisms. A micrograph is just a photograph of a microscopic image. Dark field is when the specimen is made to appear light on a dark background. Dark field stops help prevent the light from entering the condenser. Here's a short little video on using the microscope. Another type of microscope is the phase microscopes. One type is phase contrast. These can be used to examine living organisms that would be damaged by affixing them to slides or staining. We use two sets of light rays. It's bright where they are in phase and dim where they are out of phase. This can be useful for observing flagella and cilia. Differential interference contrast microscope, or Nemarsky, creates interference patterns. It splits the light into its components. Increased contrast is shown, and then it will show a three-dimensional image. Fluorescent microscopy will use a UV light source to fluoresce the objects. This has a shorter wavelength and increased contrast because of the dark background. Fluorescent dyes are used. This can be used with organisms like Pseudomonas aeruginosa, Bacillus anthracis. Confocal microscopes use fluorescent dyes or antibodies and UV lasers to illuminate a single plane. These are no thicker than 1 micron, so the slices get digitized on a computer to create a three-dimensional image, much like the way you see the slices of images used in an MRI. TPM is two-photon microscopy. This uses long wavelength red light. Two photons are needed to excite the fluorochrome to emit light. This allows us to have images of living cells and tissue up to 1 millimeter deep. The scanning acoustic microscope. This interprets the action of sound waves sent through a specimen. The resolution of this is about 1 micron. This can be used to study living cells attached to another surface like cancer cells or biofilms. Electron microscopy. Here the wavelengths are going to range from 0.01 nanometers to 0.001 nanometers versus the light microscope using wavelengths of 400 to 650 nanometers. That allows us to magnify the image between 10,000 and 100,000 times. There's two main types of these microscopes. The transmission electron microscope, or TEM, generates a beam of electrons for an image on the screen. It's going to be enhanced using electron-dense stains. These have to be used in a vacuum, so in addition they are to being toxic, there's the inconvenience of having to prepare them in a vacuum. The specimens have to be very thin, dehydrated, and put in plastic. And this works up to about 100 nanometers thick. The scanning electron microscope, or SEM, here the electrons are used to view the specimens on the surface. This will magnify about 10,000 times with 20 nanometer resolution. The whole specimen can be observed, and again it's going to need a vacuum. Here's a video clip showing a couple of variations of the electron microscope. Probe microscopy is relatively new. It uses pointed electron probes to magnify things up to 100 million times. So the STM scanning tunneling microscope, here you have a flow of current between the probe and the specimen. The specimen has to be electrically conductive, and this will allow you to view the surface. It can measure things as small as 0.01 nanometers. The atomic force microscope, or AFM, measures the deflection of a laser beam. These can magnify specimens that do not conduct electricity. They can be living and no vacuum is needed for these. Here's a brief video clip on these types of microscope. Staining is really important with microscopy. We color the specimens with various dyes or stains. Your smear is the thin film of the organism that is placed on the slide. When people first start doing things in microbiology, sometimes there is a tendency to create smears that are relatively thick. A thin smear is going to be more useful. It will actually allow you to see the entire cell. If the smear is too thick, you have multiple cell layers on top of each other. The organisms need to be fixed to the slide. This can be done using heat or chemicals. Using heat, the slides are gently heated. They pass through the flame with the smear up. Chemicals such as methanol or formalin can be applied for 1 minute to help fix the slides as well. When we look at basic principles of staining, we have acidic dyes and basic dyes. Acidic dyes have an anionic chromophore. They'll stain alkaline structures. Basic dyes have a cationic chromophore. They'll stain acidic structures. Basic dyes are more common because most cells are negatively charged. Simple stains are going to be composed of just a simple basic dye. These include crystal violet, safranin, and methylene blue. With differential stains, you use more than one dye, so different cells or chemicals or structures can be differentiated when looking at them underneath the microscope. The gram stain is a very common one in microbiology. It differentiates between two large groups of organisms. This was developed in 1884 by Hans Christian Gram. With this procedure, you start out with your primary stain. It's going to be crystal violet placed on the smear for 1 minute. The mordant, like iodine, it's sometimes referred to as Gram's iodine, is a solution that's placed on for 1 minute. This makes the dye less soluble. The third step is to decolorize. Here you rinse with ethanol and acetone, and you rinse the solution for 10 to 30 seconds and then with water. This is one step where sometimes people will get a little bit carried away and actually rinse the organisms clear off the slide. Finally, you use a counterstain. Safranin is placed on the slide for 1 minute, and then it's rinsed with water and blotted dry. This image here is an example of some gram-positive and gram-negative cells. Gram-positive cells stain purple, and the gram-negative cells stain pink. Some stains are going to have waxy cell walls, so they don't stain with simple stains. For these, we would use an acid-fast stain. This would include organisms like Mycobacterium and Nocardia. The Zeil-Nielsen procedure starts out where you cover the smear with tissue paper to help retain the dye. Next, you would flood with carbofusion for several minutes, and you warm it over steam to help drive the stain into the waxy wall. You basically are helping to melt the waxy wall. Next, you remove the tissue paper, cool, and decolorize with hydrochloric acid with a pH of less than 1. Only the cells with wax will retain the stain. Then you counterstain with methylene blue. This is an example of an acid-fast stain, and then there's a video clip of an acid-fast stain. You'll be doing these in the lab in some cases. Endospore stains. This would be Bacillus and Clostridium. That would be examples of these. Here you use the Schaefer-Fulton method. This is used to drive the malachite green into the endospore. This is done after cooling, and then you would decolorize with water and counterstain with safranin. This will allow you to end up with green endospores and red vegetative cells. This is an example of an endospore stain. A negative stain, or a capsule stain, stains the background and then leaves the cell colorless. Eosin and nigrosin are going to reveal a negatively charged capsule as a halo around the cell. So you can see the halos around these cells in this picture. This is an example here of a capsule stain in this video. Flagellar stains are going to use ferrocyanine and carbofusion and other mordants like tannic acid and potassium aluminum. These are used to make the flagella appear larger and more visible. It's almost like putting mascara on the flagella to make it appear thicker and more visible. This is an example of a flagellar stain in this video. The electron microscopy stains will contain heavy metals like lead, osmium, tungsten, and uranium. So microbes also have their own form of classification. Taxonomy is the science of classifying and naming organisms. We're going to use a form of the Linnaeus method. This was developed in 1753 by Carolus Linnaeus. His contribution was important because prior names were descriptive and varied scientist to scientist. At least with the Linnaeus method, they were going to have the naming based on some common characteristics. We use a binomial nomenclature with a specific epithet. So binomial means you're going to use genus and species. With taxonomy, there are multiple groups. You have the domain, kingdom, phylum, class, order, family, genus, and species. Each as you go down from the domain to the species, each is going to get more and more specific. All the taxa have Latin names. The species has a descriptive name that consists of the genus and the specific epithet. So the genus name is usually a noun and is capitalized. The species is usually an adjective and going to be in lowercase, and both of them would be in italics. When we look at the classification, we have three domains of life: the eukarya, bacteria, and the archaea. These are all based on the types of ribosomal nucleotides. When we look at the cell types, we have two cell types, the prokaryotes and eukaryotes. The domains of life are going to include the bacteria and archaea, which are both prokaryotic cells, and the eukaryotes, or eukarya, are going to be eukaryotic cells. We have several identifying characteristics that we look at with microbes. Your physical characteristics will include the morphology or shape. We can do differential testing for this. Biochemical tests are relied upon very heavily. These are used to identify things when they appear similar. If you look at gram-stained organisms, there's several organisms that are going to have the exact same appearance on a gram stain. In those cases, a biochemical test would be very useful to determine the taxonomy. Serological tests can be used. These are agglutination tests. You test for the presence of an antigen using an antibody. Antibodies are very specific to the antigen, so these are usually very reliable. With phage typing, we know phages are specific for a particular host. You can use a phage to determine if a particular host is present by seeing if it actually infects that cell or not. Nucleic acid analysis, this allows us to compare nucleotide sequences. When identifying an unknown organism, dichotomous keys are used. These are a series of paired yes or no statements that help you efficiently identify an organism. So for example, you could say, does it stain as gram-positive? If yes, you're going to go down one pathway to identify it. If no, you go down another pathway. Berge's manual has been used for years. It contains information on laboratory identification of the prokaryotes. When it comes to classification of viruses, Linnaeus did not know the viruses even existed at that point, so they were not included in his original hierarchy. Virologists will classify viruses into families and genera, but the higher taxa are poorly defined. There are other means of microbial identification. Rapid identification methods are becoming more and more common. They're manufactured for groups of medically important bacteria like the enterics. These perform several biochemical tests simultaneously for identification in 4 to 24 hours. Typically, a culture is going to require at least a day or two, so these are much quicker. These use numerical identification because each test is assigned a number, and then you can use a computerized interpretation. Fatty acid profiles are another way of identifying organisms. These are commercial systems that will separate and compare the fatty acids. These can be used in clinical and public health labs. Flow cytometry allows us to identify bacteria without culturing. Here, a moving fluid with the bacteria is forced through an opening, and the presence of the bacteria is detected by a difference in the conductivity of the cells in the medium. These can also be illuminated by a laser or with fluorescence. We can use DNA base composition to identify organisms. This will also help us to draw conclusions about relatedness. The more closely related, usually the more close the DNA base composition. With DNA fingerprinting, you use restriction enzymes to compare base sequences. You compare the size and number of restriction fragments. They're sometimes called riplips. This gives information about the genetic similarities and differences and how closely related the organisms are. The NAATS, these are nucleic acid amplification tests. These can be used to increase microbial DNA levels that can be tested by gel electrophoresis. These start out using PCR or RT-PCR or real-time PCR. So with these, you can have very small amounts of information using PCR to amplify the genetic information and then analyze it. Cladograms are a tool that will create a map, and these show evolutionary relationships. With the nucleic acid hybridization identification techniques, here DNA strands are heated and separated. These are going to measure the ability of a DNA strand from one organism to hybridize, which is when you rely on the complementary base pairing, with a DNA strand of another organism. So the greater the hybridization, the greater the degree of relatedness. This is the basis of several techniques that are used. With southern blotting, you use DNA probes to test for hybridization of DNA strands. So you would have a DNA probe, and then you would look to see if it would hybridize with various DNA strands to identify things. DNA chips or microarrays are these chips composed of DNA probes. The sample or unknown DNA is added to the chip with a fluorescent dye, and then you look to see where did it hybridize. You're looking for hybridization between the probe and the DNA sample, and you can identify it based off of which sample it hybridized to. With ribotyping and ribosomal RNA sequencing, this allows us to determine phylogenetic relationships. We can use PCR, the polymerase chain reaction, to amplify signature sequences and then compare the restriction fragments on a gel electrophoresis. The ribosomal RNA sequencing is useful for the phylogenetic relationships, especially in the eukaryotic world, where those are generally passed on through the egg cell. FISH, or fluorescence in situ hybridization. Here the fluorescent dye labels RNA or DNA probes. These can be used to stain microbes in place, which means they can be left in the environment or in the patient, and this will allow you to get results without having to wait for a culture.